Okay, Dr. Gallardo is from the Washington University in St. Louis, Missouri. I think he is one of at least four research grants that we've given to this fine institute. And he received a $100,000 grant in 2021, and his CART grant was to look at drugs that deal with inflammation that affected the brain cell neuropathy, focusing primarily on the tau tangles. I think I got that right. Dr. Gallardo received his Ph.D. in 2008 from the University of Texas Southwest Medical School in Dallas, in the laboratory of Dr. Thomas Sudhoff, now at Stanford University Medical School, where his thesis focused on studying, and bear with me on this, acyanonuclein, a presynaptic protein that has been implicated in Parkinson's disease. And he went on to get his postdoctoral studies at Harvard Medical School, where he focused on the mechanisms behind amyotrophic lateral sclerosis, which we all know as ALS. And I'll tell you, this really hits a nerve, Dr. Gallardo, because my wife's sister died of ALS at a very young age, and it was just an awful, awful experience. Anyway, Dr. Gallardo went on to join the faculty and laboratory of Dr. David Holtzman as a senior research scientist at Washington University School of Medicine in St. Louis in 2014, and was appointed to the faculty as an assistant professor of neurology in 2017. And there he was focusing on neuroinflammation in Alzheimer's diseases. And that's about the best I can do. I'm going to turn it over to you and let you give us the details. Thank you very much for being a researcher for CART, and thank you for joining us. We appreciate you taking your time, letting us know how well you're doing and what you're doing. Dr. Gallardo, the floor is yours.
Thank you so much for the invitation and also the introduction. It's like I said, it was a wonderful experience being part of the CART Fund. So today we're going to be talking about astrocytes. That's probably most of you have heard of the other glia cell, which are the microglia, and we'll briefly introduce the microglia and some of the pathologies that we are associated with Alzheimer's disease. Alzheimer's disease is considered a dual proteinopathy, in that it's characterized by the accumulation of these extracellular amyloid plaques. Now, these amyloid plaques are composed of these amyloid beta 42 peptides that are synthesized from this APP, amyloid precursor protein. In addition to the accumulation of amyloid plaques, we also get the accumulation of intracellular tau tangles. Now, tau is a protein that is usually confined in the axons of healthy neurons, along with the microtubules. However, as the disease progresses, this tau protein becomes hyperphosphorylated, which liberates itself from these microtubules and tends to aggregate in the cell body. Now, there are some key features that are different between tau pathology and A-beta, and predominantly that tau pathology associates closely with the progression of disease. So more dementia, more degeneration, more tau pathology. That's not necessarily the case with A-beta plaques. A-beta plaques can accumulate many years before disease onset, and the switch between it becoming detrimental and how it actually contributes to neurodegeneration still remains unclear. However, we do know that A-beta plaques have a direct role in Alzheimer's disease. There are mutations in the APP gene that are associated with familial Alzheimer's disease, and these patients tend to get AD, dementia, much earlier in their 50s. Now, the relationship between A beta and tau is still unclear, but there is another component that contributes to both A beta plaques and tau pathology, and this is brain inflammation. Now, some people call it neuroinflammation, brain inflammation, and it's a really loose term because we don't really understand the key factors in inflammation. There's a number of cytokines and chemokines that are being expressed, but we do understand the key players. And one of them is the microglia. Now, microglia recently has gotten a number of attention, and there's been a number of studies showing that microglia play a direct role in somehow regulating the amount of A beta plaques. What has gotten less attention, though, is the other glial cell, which are the astrocytes. Now, I will refer to a reactive astrocyte as an inflammatory astrocyte. This is when they become stimulated, and they become stimulated to A beta plaques and also to the accumulation of tau tangles. And our research is aimed to try to understand the mechanisms that regulate the inflammatory response of astrocytes, in that we have shown and others have shown this inflammatory response progresses both tau pathology and also A beta plaques. So with that, I want to give a little bit of a history about the glial cells. The term glial cell was first introduced in 1858 by Rudolf Virchow in a series of lectures at a medical school. And this is one of the drawings he had back in 1850s, 1858. And what basically he was describing are these other cell types that were among the neurons, and referred to as these as glial cells, or the glue of the central nervous system, in which he proposed the idea is that these glial cells are basically the scaffold for the central nervous system, enabling the neurons to make connections among themselves and make the synaptic activity. Now, shortly following his description of these glial cells, a number of anatomists started drawing these glials from 1862 to the early 1900s. And what was known really early on is that their morphology was quite dynamic. Now we know there's actually three different glial cells. There's the oligodendrocytes that are important for regulating the myelin sheaths, the astrocytes, and also the microglia. And this drawing here is from Golgi. It's a really remarkable drawing. So what you see is these are the actual astrocytes. And what we knew early on is that they made really nice connections with the blood vessels. All along the blood vessels in the brain, we have these astrocytes making connections, which we call the end feeds. Now, the term astroglia first actually was introduced in 1893 by Michael von Leeuwenhoek. And basically, he was an anatomist, and he said these glial cells look like star shape. We should probably name astrocytes. And astrocytes was first coined in 1893. In 1910, the first indications that potentially astrocytes or glial cells have a role in neurodegenerative diseases came from the infamous Dr. Alois Alzheimer himself. Now, he published this book that is lesser known, in which he drew these glial cells along neurons that had the accumulation of tau pathology. We now know that this aggregate inside the neurons is tau pathology, and these are likely either microglia or astrocytes that he was looking at back in 1910. Now, this is a brief history of astrocyte biology and some of the key findings. Now, this is my own personal view. There's probably a number of other ones, but I think what is really highlights here is that there's far and few major discoveries in astrocyte biology. Since their first description in 1858, it was just commonly thought that these were just a scaffold for the central nervous system. And it wasn't until 1958, 100 years later, that Dr. Held, who was a physiologist, was able to record from these astrocytes, suggesting that they had other biological functions other than being a scaffold, and that they're likely regulating ionic homeostasis. In 1963, it was shown that astrocyte RNA changed in Parkinson's disease. They further found that they made these gap junctions are similar to synaptic junctions. And in 1965, they showed that this astrocytes regulate the sodium and potassium. Now, this was a key finding that sodium and potassium plays a major role in the central nervous system. And I'll talk a little about that a little bit later on. The next major discovery came in 1972, in which they identified GFAP. Now, I'm going to use GFAP as a common marker for astrocytes throughout the talk. This was the first protein that was enabled us to start studying astrocytes in the brain. Shortly afterwards, it was found that astrocytes were responsive to brain injury. This was just a stab wound in a mouse model, and they found that the astrocytes GFAP protein went up and surrounded the brain injury. And then throughout the 1980s and 90s, the major finding was that these cells were pro-inflammatory. They're responsive to chemokines, cytokines, endotoxins, and had very characteristics to an immune cell in the periphery. The last thing I want to highlight is this 1997 study. It was first described in 1904 by Golgi that the astrocytes made these contacts with the blood vessels, and it wasn't until 1997 that they actually showed the biological function of astrocytes regulating the blood-brain barrier. It almost took 100 years. And then we jump into the modern day era of astrocytes. Now, we started studying astrocytes in a serendipitous discovery. As mentioned earlier, I was looking at familial ALS. ALS is a devastating disease. It's a motor neuron degenerative disease. The lifespan is between 3 to 5 years after disease onset. And what we discovered is that there is an upregulation of this alpha-2 sodium ATPase that was predominantly expressed in astrocytes. Now, these are human ALS patients, and looking at a mouse model, we found that this was also elevated. The sodium ATPase plays a key role in regulating ionic homeostasis. It pumps in 3 sodiums into the cell and pumps out 2 potassiums outside the cell. And what that causes is this concentration gradient of sodium and potassium. This is called the membrane potential. And a number of cells in the central nervous system utilize this membrane potential to produce physiological functions. And one key function is what I'll go into later on. But finding there was an increase of the sodium ATPase, we decided to dive a little bit into what is the potential function of the sodium ATPase in ALS. And we took our mouse model and we crossed it to a heterozygous where we knocked down the sodium ATPase. This is a global knockdown. And what we found is that we can increase their overall survival. And if we knocked it down in the spinal cord, in red are our motor neurons, in green are the astrocytes in which we knocked down the sodium ATPase. We found that there was an increase in motor neuron survival. Looking at areas where we don't knock it down, we see that the motor neurons are significant atrophy and a lot less survival, whereas knocking down the ATPase in astrocytes led to increased survival and preserving the motor neuron atrophy. As I transition into getting my own investigation in the laboratory, the question was, is this sodium ATPase playing a role in other neurodegenerative diseases? And the first disease we decided to look at is Alzheimer's disease. Now, looking at human brain samples, this is public RNA-seq data from a number of different studies. And we found that very similar to an ALS, there was a significant increase in the amount of alpha-2 sodium ATPase gene expression. Looking at brain samples from human AD patients and controls, we found that, again, there's an increase of sodium ATPase that correlated with an increase of this GFAP protein, the one that is a marker for astrocytes. And looking at our mouse model that develops the tau pathology, we saw an age-dependent increase of the sodium ATPase together with an age-dependent increase in GFAP protein. So the question was pretty evident from the get-go. If we target and block the sodium ATPase in mouse models of tauopathy or amyloidosis, can we suppress the astrocytic response and decrease plaque burden and tau pathology? And to do so, we took 2 different distinct approaches. In the first approach, using a tau mouse model that accumulates tau pathology in an age-dependent manner, at 8 months, these mice develop pathological tau and also brain inflammation. And we decided to block the activity of sodium ATPase pharmacologically using a drug known as digoxin. And we infused digoxin directly into the brain for 6 weeks. Looking at the amount of tau pathology, this is dark staining here, we found that this led to a reduction of tau pathology throughout the hippocampus. And also, these mice developed significant brain atrophy. We see enlargement of the ventricles and thinning of the peripheral cortex. Infusing digoxin, blocking the sodium ATPase activity, led to a protective effect, preserving this brain atrophy. In our mouse model for amyloidosis, we took a different approach. We took a genetic approach. And we took a mouse that develops these characteristic amyloid plaques, which is seen in Alzheimer's patients, and we crossed into a mouse in which we can do conditional knockout of the sodium ATPase selectively in astrocytes. We aged our mice to 8 months. Again, this is after disease onset. Did the conditional knockout of the sodium ATPase in astrocytes. And what we found is that this led to a rather drastic decrease in the amount of plaque pathology when we knocked down the sodium ATPase. Looking at these astrocytes, this is GFAP. Around the plaques, you see that there's an abundant amount of GFAP. When we knock down the sodium ATPase, we see that this decreases the amount of GFAP protein around the plaques. This is also evident in our tau mice. This is our control wild-type mice. This is our tau transgenic mice. And this is after blocking the sodium ATPase. We see that there's a decreased amount of GFAP protein, which is quantified here. But we also see a decrease in microgliosis and also a number of pro-inflammatory cytokines and chemokines that have been found to be elevated in human patient samples. So what this suggested early on to us is that if we block the sodium ATPase, we can then, in astrocytes, either by conditional knockout or by pharmacologically, we can suppress their inflammatory response, decreasing tau pathology, also the accumulation of amyloid beta plaques. However, one major caveat about the sodium ATPase is that it is expressed in the heart. And doing anything pharmacologically that targets the heart is probably something that's incredibly challenging. So it was important for us to try to identify a mechanism to how the sodium ATPase is potentially regulating this inflammatory response. This will enable us to develop potential therapeutics that are selective for astrocyte inflammation. And the hypothesis that we decided to chase is that one major function for the sodium ATPase is regulating the sodium-potassium gradient. And one biological function that utilizes sodium are amino acids to be transported into the cell. Now, SNAT transporters will couple sodium with neutral amino acids, such as glutamine, transport it inside the cell, increasing the amount of sodium levels intercellularly, and the pump will pump out the sodium outside, regenerating the sodium-potassium gradient, increasing the amount of glutamine. Now, we decided to focus on glutamine in that in the periphery, it has been well established that glutamine plays a role in inflammation. Blocking glutamine into T cells, B cells, and other inflammatory cells peripherally blocks their inflammatory response and attenuates inflammation. Based on those studies, we thought potentially the sodium ATPase is increased to increase the amount of glutamine uptake into astrocytes, and that potentially may be contributing to inflammatory response. And to address this question, we took our mouse model for our amyloidosis that developed this characteristic amyloid plaques, looked at SNAT2, which is a transporter for glutamine, together with our GFAP marker, and we found that there is a robust increase of SNAT2 expression around the plaques. Looking at AD human patient samples, looking at around the plaques, again, there's an increase of SNAT2 that co-localize with our astrocytic markers, suggesting that there's an increase of SNAT2 together with an increase of sodium ATPase. Now we can also look at these astrocytes by taking astrocytes from the brain of a postnatal mouse and putting them on a dish. And what this gives us is a way to look at more cell biology in the dish. And the first thing we need to do is to determine whether these astrocytes are characteristic to the astrocytes that we see in our mouse model in human patient samples. So we stimulate our primary astrocytes with a cocktail of cytokines. Now, these are just pro-inflammatory molecules. And we see that there's an increased amount of SNAP2, an increased amount of sodium ATPase, and our GFAP marker, suggesting that they are mimicking the response that we see in our mouse models and in AD patient samples. The first question we wanted to address was, are we increasing the amount of glutamine inside the cell? So using our primary culture, in which we can stimulate these with cytokines and chemokines, and using the glutamine that's fluorescently tagged, we can measure the amount of glutamine inside the cell. This is in the absence of the tagged glutamine. This is our control astrocytes. We see that there is an increase of glutamine under basal conditions. When we stimulate these, we see there's an increase of glutamine uptake. And When we block the sodium ATPase, we see that this decreases glutamine uptake, as well as when we block the transport, suggesting that reactive astrocytes increase glutamine that are codependent on the SNAP protein and also the sodium ATPase. But do they actually play a contribution in pro-inflammation? To address this question, we look at a number of pro-inflammatory gene expressions in our primary culture dish. This is our control conditions. This is activating them with these cytokines. We see a robust increase in pro-inflammatory gene expression. Blocking the SNAP transporter or blocking the sodium ATPase, we see that we then suppress this pro-inflammatory response, suggesting that glutamine uptake plays a key role in the inflammatory response. Now, glutamine is a really unique molecule, amino acid, in that it can also work as a signaling molecule, but it can also be metabolized. In the central nervous system, glucose and glutamine are the most abundant metabolites. Now, glutamine can be metabolized into the TCA cycle by a process known as glutaminolases. And there's two key enzymes that metabolize glutamine. The first one is this GLS2 enzyme that will metabolize glutamine to glutamate. And glutamate will further be catabolized to alpha-ketoglutarate by GLUD enzyme. And alpha-ketoglutarate can then go into the TCA cycle for energy production in the mitochondria. Our question was, once inside the cell, is glutamine being metabolized or is it being used as a signaling molecule? Again, going back to our mouse model and our human patient samples that have the amyloid beta plaques, we looked at the GLS2 enzyme and GLUD enzyme in astrocytes. Now, GLS2 is in neurons. The staining back here in our control wild type is neurons, and we do see it in the neurons in our ape mouse model for amyloidosis. But what you see here is along where there's GFAP, there's an upregulation of the GLS2 enzyme that metabolizes glutamine to glutamate. This is also evident in our human patient samples. GLS2 is increased in astrocytes around where there's near around the plaques. Looking at our second enzyme for glutaminolases, which is GLUD, that produces alpha-ketoglutarate. Now, this is a punctated one because this is actually the mitochondria. Again, around the plaques, we see that along the astrocytes, it's GFAP, we see that there's a robust increase of GLUD enzyme. It's quantified here, and we also see this in our human patient samples, suggesting to us that potentially, once the glutamine is gained inside the cells of astrocytes, this is then going into the TCA cycle for energy production. And to address this question, we can take again our primary cultures in a dish, stimulate them with cytokines, and very characteristic of what we see in our human patient samples and our mouse model. We see that there's an increase of GLS2 enzyme, increase of GLUD, an increase of our SNATs, sodium ATPase, and our GFAP protein. Now, once inside the cell, we can actually trace glutamine, in which we can use a carbon-labeled glutamine. And we can look at to see inside the cell, where is it going as far as metabolites, using a mass spec analysis. So stimulating our astrocytes, adding radio-labeled glutamine into the conditioned media, collecting the astrocytes, we found that this is our control. Astrocyte cultures, stimulating with cytokines, we see there's a robust increase in the amount of glutamate label, suggesting that glutamine is being catabolized to glutamate. And we also find that there's a significant increase in alpha-ketoglutarate, suggesting that glutamate is further being catabolized to alpha-ketoglutarate. Furthermore, we see virtually all of the TCA cycle metabolites being enriched, suggesting that astrocytes are increasing glutamine uptake for energy production in the mitochondria. What happens if we block glutaminolases? Will this actually affect the pro-inflammatory response? And we can address this question by using a pharmacological approach. Taking our astrocyte cultures on a dish, BBTS is a selective inhibitor of GLS2 enzyme. So this will block the conversion of glutamine to glutamate. Stimulating with cytokines, we see that this led to a decrease in GFAP protein, a decrease in the GLS2, GLUD, which is quantified here. R162 is another pharmacological agent that blocks glutamate to alpha-ketoglutarate production. Very similar to what PBTS or GLS2 inhibition does. We see that there's a really nice suppressive effect of the enzymes that metabolize the glutamate inside the cell. Now, looking at the pro-inflammatory response, this is just our cytokine stimulated astrocytes. We see that there's an upregulation of pro-inflammatory genes. Blocking GLS2 enzyme, we see that there's a suppressive effect of pro-inflammatory genes, or blocking the GLUD enzyme, which produces alpha-ketoglutarate. We see that there's a suppression of pro-inflammatory genes as well. So just as a brief recap, what we have found is that in Alzheimer's disease and also in patients with ALS, there's an increased amount of glutamine transporter SNATs, increased amount of the sodium ATPase, and the SNATs and sodium ATPase are codependent for the transport of glutamine. Now, once inside the cell, glutamine can be metabolized by glutaminolases by enzymes such as GLS2, which converts glutamine into glutamate, and GLUD, which converts glutamate into alpha-ketoglutarate for the TCA cycle utilization. And if we block either the transport of glutamine or we block its metabolism, we can suppress the reactivity. And what we've shown, at least in culture, this leads to a decrease in pro-inflammatory response. But the main question, what does it do to the amyloid pathology in our mouse model? Now, to address this question, we used a CRISPR-Cas9. Now, CRISPR-Cas9 is a phenomenal tool that we can use to knock down genes of interest. And we combined that with AAVs, adeno-associated viruses. Now, AAVs have become another fabulous tool for studying CNS, but it's also become therapeutic. There's a number of diseases that are now being treated with AAV gene therapy, including spinal muscular dystrophy and also blindness in human patients. So what we did is we combined AAVs with the CRISPR-Cas9 approach, in which we can selectively target the knockout of GLUD enzyme. Now, as you notice that along the pathway of glutaminolases, we also generate glutamate. But one key aspect is that the glutamate generated from glutamine is different from the glutamate we get exogenously. So if we would deprive cells from glutamine, we cannot rescue them by giving them excess glutamate. What that suggests is that glutamine metabolism is independent from glutamate that is exogenously uptaken. That doesn't mean that glutamate doesn't play a role, but we can then target GLUD enzyme that metabolizes, is the last step in our glutamine metabolism, and ask the question, does that suppress the activity of astrocytes in vivo? Looking at our mouse model for amyloidosis, we age these mice to 8 months. This is after disease onset. We knock down GUD specifically in astrocytes, age them another 4 months to 12 months. Look at the amyloid plaques, and we see that there's, in our control mice, compared to our knockout approach, we see that there's a decreased amount of amyloid plaques, decrease of GLUD enzyme. And looking at our GFAP protein, looking around the plaques, we see that this leads to a significant amount of decrease of astrocytes around the plaques, both in females and males. Looking at fibrillary plaques, we see again significant decrease in plaque burden in both males and females. Another hallmark of Alzheimer's disease are what's called dystrophic neurites. Now, this is an accumulation inside neurons with a LAMP1 protein. LAMP1 is a lysosome marker. And around the plaques, you see this accumulation of LAMP1 that is a hallmark for neuronal distress. When we knock down the GLUD or suppress glutaminolases in astrocytes, we see that there's a really nice significant decrease in the amount of dystrophic neurons. Actually, this is the females. I missed to put the male graph here, but it's both significantly decrease in both males and females. Now, more recently, there's been some suggestions that astrocytes are not uniform, that there's potentially different types of astrocytes in the brain. To address that question, we use a second marker for astrocytes, which is S100 beta. It's also increases in AD patients, in ALS patients, and also in Parkinson's disease. Just looking at the amount of S100 beta, it's significantly increased when we knock down the GLUT enzyme and selectively in astrocytes. We then see a significant robust decrease in the amount of S100 beta. This suggests that the glutaminol lasts in the pro-inflammatory response is not selectively to only astrocytes that express GFAP, but they're also to astrocytes that are expressing S100 beta. So at this point, we've been able to show that there's an increased amount of the sodium ATPase, which regulates the sodium-potassium gradient, an increased amount of SNAP transporters, which transport glutamine inside the cell. And inside the cell, glutamine is metabolized through the TCA cycle. And if we block these pathways, we can suppress the reactivity in mouse models of amyloidosis, ALS, and also in tauopathy. So that's 3 different distinct mouse models of proteinopathies. But one of the key features that really got our attention is that glutamine is a conditional essential amino acid. And what that means is that the body can actually generate glutamine. Peripherally, the liver makes a significant amount of glutamine. But under conditions of metabolic stress, dietary glutamine plays a key role, particularly in peripheral inflammatory diseases. The uptake of glutamine from diet is necessary for immune cell responses. Now, in the central nervous system, we kind of ran into a little conundrum. The cells that produce glutamine in the CNS are actually astrocytes, and they generate glutamine from glutamate. So they convert glutamate into glutamine. But our studies are showing that they also utilize glutamine for reactivity. But they also raise a second question is, does dietary glutamine actually play a role in brain inflammation and astrocyte biology? And as we embarked into this question, which is a really new direction for our lab, the question became actually more relevant. Now, all of us here are actually well aware that Western diets are associated with metabolic diseases. But most of us also hear that they're associated with carbohydrates, sugars, trans fats, and fats. And what's lesser known, though, is that Western diets also increase dietary protein. Now, the U.S. Institute of Medicine recommends that men take around 56 grams of protein, whereas females take 46 grams of protein. And it turns out the Western diets actually increase the amount of daily dietary protein, almost double. Around the age of 30, that's around 100 grams for men, and women will take around 70 grams, and kind of say that consistently. That's 1.5 to 1.7 times more dietary protein in Western-based diets. And when we look at the amino acids, this study here, compared that to what they would call to metabolically healthy diets, such as vegetarian and the Mediterranean diet. Looking at the Western diet, there's a significant increase in virtually all amino acids. Now, more recently, it's been shown that high protein diets are actually adversive. And single amino acids can also be adversive. And if you manipulate amino acids, you can actually decrease the amount of tumorigenesis. Or if you increase the amount of branching amino acids, this actually leads to a decrease in longevity. It has also been associated with cardiovascular disease, diabetes, and more recently, there was 2 studies out, so they didn't put it in more recent. They found that high protein diets exacerbates A-beta pathology, and decreasing low protein diets can potentially be protective. But the main question is, of the 20-some amino acids, which ones are contributing to these diseases, including the amyloidosis and AD-related pathologies? So we decided to ask a question, what if we increase dietary glutamine? Is that going to be adversive? So we took mice that are 6 months, that are amyloid plaque producing mice. Now this is before disease onset. There's no plaques, there's no brain inflammation. And we gave a high glutamine diet for 2 months. Looking at the CSF, this is the cerebral spinal fluid taken from the the little mice, we see that there is a significant increased amount of CSF glutamine in both the females and males. Now, in the plasma, it's a little bit different. Females have a significant increase in plasma, whereas males do not. Now, we think this is predominantly due to the males having more muscle mass, and muscle uses a significant amount of glutamine. But we do have some other hypotheses of what we can potentially investigate this phenomenon. But what's really surprising to us is when we increase the amount of CSF glutamine levels, looking at the amount of astrocytes, these astrocytes were exacerbated. There was a significant amount of astrocytes compared to our control diet. And we see that there's a significant amount of A beta plaques. This is the amount of astrocytes or control diet in both females and males. Looking at total plaques, both females and males, we see a significant increase in total plaques in the cortex. In the hippocampus, it wasn't quite significant in males. Now, we do have some hypotheses about why we're not seeing too significant in the males. This is a young cohort. This is 8 months. Fibrillary plaques, which is another marker for a more adverse type of amyloid plaque. Again, the females and males have a significant increase in the cortex. In the males, we don't see too much of a significant increase, but we do see it in females. Now, one unique aspect about the female mice is that they're more aggressive in producing amyloid beta plaques. Looking at our dystrophic neurites, our control diet, compared to our high glutamine diet, we see there's an increase of dystrophic neurites in both males and females. And this finding here in microglia is incredibly puzzling to us. We actually see a decrease in females in a high glutamine diet and an increase in males. We don't quite understand what is going on. The only consistent thing we find is that there's an increased amount of vascular gliosis together with an increased amount of amyloid beta plaques. There's still quite a bit of questions we need to address with the microglia. Now, as we all know that Alzheimer's disease is, we're getting better at being able to diagnose it, but most of the time we diagnose Alzheimer's disease after disease onset. So it's important for us to address the question what if we increase dietary glutamine after disease onset? So we age our mice at 12 months. These little mice now have plaque pathology, brain inflammation. Gave them a high glutamine diet for 2 months. And again, there is a robust increased amount of astrogliosis, a significant amount of astrogliosis in all brain regions in both the males and females. Looking at total plaques, there was a significant increase in virtually all brain regions in both males and females. Looking at the fibrillary plaques, again, there is a robust increase in virtually all brain regions. Some of the males didn't reach significance in some of the brain regions, but was a strong trend in the increase in A beta plaque pathology. Looking at our second marker for astrogliosis, we find that this also led to a significant increase of S100 beta in the males and females in pretty much all brain regions. And looking at dystrophic neurites, again, there is a significant increase in both males and females in virtually all brain regions, suggesting that increasing the amount of glutamine in the central nervous system seems to aggravate these astrocytes, leading to a more profound inflammatory response and increase in A beta pathologies. So just for a recap, these are the findings we have so far. We have found that reactive astrocytes increase the sodium ATPase, together with an increase in those SNAP transporters that increase glutamine transport. And the sodium ATPase and the SNAPs are codependent on increasing the amount of glutamine inside the cells. Activated astrocytes increase glutamine uptake and utilize that for TCA cycle metabolism. And if we either block the SNAP transporters, block the sodium ATPase, or the utilization of glutamine, we can suppress the inflammatory response and decrease A beta pathology in our mouse models. One intriguing fact that we further found is that we looked at glutamine in the brains of human patient samples and looked at glutamine in the brains of our IPP mice. Now, this is not in the presence of a high glutamine diet. This is just age-wise. And we found that there's an increased amount of glutamine. Now, we really don't understand this data yet because this is actual total brain glutamine. This is not glutamine that is circulating freely. Our other finding is that dietary glutamine appears to play a role in progressing the amount of A beta plaques and also activating astrocytes. If we give a high glutamine diet before disease onset or after disease onset, this leads to a significant increased amount of glutamine in the central nervous system, which seems to aggravate the astrocytic response and also increased amount of amyloid beta plaques. Now, this study we are about to submit, I hope next week. It's been a number of work for Sri Darshani, which is one of our postdocs. But this also leads to a number of key questions that we want to further follow up, including what happens if we restrict dietary glutamine? Can we then suppress the inflammatory response? Or is there other amino acids that are playing a key role in progressing Alzheimer's disease? There's also, can we use pharmacological agents to block this metabolic pathway and suppress astrocytic reactivity? With that, I want to say thank you to, obviously this work is done by people in the lab that I'm incredibly grateful to have. Sri Darshani is our postdoc. She's been doing these glutamine studies together with Sophia. I wasn't able to share the studies that Aizan has been working on, but she's been developing strategies to block inflammation through a combination of gene therapy and also through some using immunotherapies. And our former members, collaborators, and obviously I'm thankful for the CART support and this opportunity to share our ongoing works. And my Maine Coon and Ragdoll are my emotional support platform. Happy to take any questions.
Questions, anyone?
Can we go back to the full screen?
Sure.
I mean, we need to go back to where we can see everybody.
Oh, okay.
Thank you. Tell me in simple terms, if you would please, diet and amyloid beta. What specifically are the recommendations at this point?
It's a new area, I would say that. What has been shown is in patients, Mediterranean diets that have a decrease in protein intake, it's around 50% less than Western diets, has been shown to decrease the rate of dementia in patients. But diet is incredibly complicated, so we don't exactly know what combinations of amino acids, fats, and saturated fats and sugars are playing a role. So this is why our study is going to be quite significant, because it's going to be the first one to illustrate that a single amino acid may actually be contributing to inflammation and in the brain. The other thing that has been shown is that high protein diets that were advocated, all of us have probably heard the Atkins diet, that does lead to weight loss. But it also has led to a decrease in longevity, and we don't understand why. But if you give a high protein diet to mice, they tend to die earlier. And women specifically, there's been an association with a decrease in longevity for women. And also in the development of diabetes and hypertension, which are risk factors for developing Alzheimer's. So that's a question, that's an answering saying we don't know exactly, but there are hints that dietary can modulate the progression of Alzheimer's disease.
So it's still a research area, and it sounds like you leave the protein bars alone if you're a mouse, but if you're a human, that's okay. At least that's my understanding, right?
We actually think high protein would not be is good for people. Now, it probably has to do a lot with activity. Obviously, if you're working out, you need high protein, and significant
Gotcha.
amount of protein. Uh, but if you're not working out extensively, probably high protein diet is not going to be something good.
Wow. Thank you very much. I appreciate that. When we give presentations to various Rotary clubs, they say, yeah, the research is interesting. However, what can you tell us now? And so we're stuck with, well, we don't really know. There's no drugs that have been identified as really effective, but we do know that diet is a factor, exercise is a factor, brain activity is a factor, sleep is a factor, and everything that you add as a researcher helps us tremendously because we would love to prevent Alzheimer's or delay Alzheimer's since there is no cure on the horizon yet. And with that, I'll be quiet and let other people ask questions. But thank you.
So, thank you for the opportunity. Yes.
I have a question. Is there a difference in plant-based proteins versus meats?
Oof, we've discussed that and thrown that around. We don't know, but the only main possibly the one difference would be is obviously the amount of protein content versus meat versus plant-based. But also we're not too sure whether plant-based dietary protein is metabolized slower. So if you metabolize it slower, you're going to get less circulating amino acids, whereas protein may be metabolized faster, and you get a rapid increase in free amino acids. But that's something we do not know yet. But it's a valid question, and we've discussed it quite readily.
Thank you.
We've proposed to do studies, and unfortunately, the NIH didn't fund it.
Are there other questions? Thank you so much for your presentation. Thank you for the information you provided to us, and thank you for your time. We thoroughly enjoyed your talk and enjoyed the information you provided to us, and we appreciate your support of CART. We really do.
Thank you so much for the opportunity.
I'm going to stop the recording, and if anyone else would